Dear students, do you know why fish spoils quickly? And what are the common methods of the fish preservation? So, in this very lecture, we shall be talking about the common methods of the fish preservation. And before proceeding, it is my request to you all to like and subscribe my channel Biolearnia. Dear students, before learning preservation, you must understand what you are fighting. That is what you have to fight. And you all know that when a fresh fish is taken out of the water, the clock begins to tick. That is, it is like a ticking clock. The spoilage starts. The bacteria are the clock's hands. These very bacteria, they act as the clock's hands, enzymes as the spring, and oxidation as the battery. So, preservation is stopping all the three things simultaneously. Now, what are those three things? These are bacteria, these are bacteria, these are enzymes, and the third one is oxidation. So we all know the responsible organism for the decomposition of the any kind of the component, they are bacteria and fungi. So if we talk about the bacteria, these very microorganisms, they are present on the skin, they are present in gut, they are present in gills, and they multiply rapidly at the room temperature, breaking down the flesh. And secondly, the fish's own digestive enzymes, they continue working even after death. They begin to soften and degrade the muscle tissue. And thirdly, third important thing, the oxidation. That is the fats in the fish, and especially in those very fishes which have the higher oil content. So this very fat that reacts with oxygen in air, and it turning rancid and producing off flavors. It means that this very fat turns rancid and it produces the off flavor, that the flavor changes and it seems to be stinky. And every preservation method you will learn is essentially one strategy, that is even remove or neutralize the conditions. And these three enemies need to work upon, that is which is moisture, oxygen, time. So we have to stop all these very things. We have to work on the heat, we have to work on the moisture, we have to work on the oxygen, we work on the time. So we shall be dealing every single preservation technique, that is ancient or modern, it operates on one of the four principles which I have just put on here. What are those three principles? That is temperature, moisture, that is chemistry, and the heat. Now keep this in mind. Now what happens when we lower the temperature? The bacterial activity and the enzyme activity, it slows down. So what are the targets? The target is the bacteria enzymes. So when we lower the temperature, we actually target the bacteria and enzymes, thereby it slows their activity, and thus fish spoilage slows down. Then secondly, the moisture is one of the enemies which fastens, which plays a very important role in the growth of these very bacteria and fungi. So without water, bacteria cannot grow. So we have to remove the moisture, and it acts on the bacteria. It does not allow the bacteria to grow. So the first one is we have to lower the temperature. Second important point is that we have to remove the moisture, and the third important point is we have to create a hostile chemistry, that is the salts, acids, or smoke. It kills or inhibits the microbes. So we have to make such a kind of the chemistry, we have to make such a kind of conducive atmosphere wherein the salts, acids, or smoke, or there is inhibition of the microbes to grow. So actually we are talking about the bacteria and the oxidation. So the fourth important point is to apply heat or seal. So by heating the microbes are killed, and then it also prevents the re-entry. And mostly we are talking about this very heat and seal when we go for the canning. We shall be talking about this. So actually our enemy which is targeted is enzyme and the bacteria. We kill microbes by heating and prevent their re-entry into the particular sealed content. We prevent their entry by heating and then sealing. We shall be talking about one by one the common types of the fish preservation. The first part, that is the chilling. When we talk about the chilling, it is of two types. So first we shall be talking about the short term chilling, that is wherein this very method the shelf life is less, and in this very method we keep fish at 0 to 4 degrees centigrade. We use ice or refrigeration. It slows the bacterial and the enzyme activity but does not stop entirely. It means that 0 to 4 degrees centigrade, the enzyme activity slows down, bacterial activity slows down, but it is not stopped. So the shelf life is only 1 to 2 weeks. So after 1 or 2 weeks, the fish begins to spoil, fish begins to rot, or begins to decompose. So this is why iced fish is still fresh for 1 or 2 weeks maximum. So this is the chilling, we call it as short term chilling. So when we talk about this very chilling, it is just like putting the ticking clock in a slow motion. So actually the clock is ticking, but it is in a slow motion. So fish, for example, when we have to take the fish to the smaller distances, we use this very chilling, that is the short term chilling, wherein we can put fish in some ice container, and then we take this very fish to the nearby areas. So point number one was the chilling, but it was the short term, wherein the temperature used was 0 to 4 degrees centigrade. It was for a shorter period of time, wherein the spoilage does not stop but slows down. But in the second case, that is the freezing, when we talk about this very freezing, it is a long term cold, wherein fish are stored at minus 18 degrees centigrade. It essentially halts all microbial growth. Herein, fish essentially halts all microbial growth and enzyme activity. So what happens in this very case? Ice crystals form inside the fish cells, and when done slowly, these crystals damage cells. So in this very case, the ice crystals, they form inside the fish cells, and when this is done slowly, these crystals damage the cells. So that is why thawed fish loses texture compared to the fresh fish. So that is why. Now thawing, what is thawing? That when we de-ice, we call it as the thawing. We mostly defreeze it. So quick freezing at minus 40 degrees centigrade forms the smaller ice crystals and preserves the quality far better. So there are two methods. One is quick freezing and other is slow freezing. In the slow freezing, this very cells are damaged, but in the quick freezing, ice crystals, they are formed. The smaller ice crystals are formed, and it preserves the quality far better. So shelf life here is, in this very case, is 6 months to 2 years in the case of the long term cold freezing. So if we have to keep the fish fresh and the taste has to be maintained, we have to quick freeze it. Then drying, the third important point in the case of fish preservation is drying, wherein what we do, we remove the moisture. So we were talking about the moisture is one of the enemies which helps in the growth of the microbes, which decompose a fish or spoil a fish. So this is the traditional fish smoking or drying. We shall be talking about the smoking later on. But in the case of drying, one of the oldest methods in the world is the drying. So we dry it. The fish they are spread on the mats or the racks in the open sunlight, and the cheapest widely used in the coastal areas, wherein we dry these very fish in the open sunlight, and this is one of the best ways to preserve the fish. The second important point is the smoke drying, that is it combines the heat and smoke. So it is a combination of the heat and smoke, and wherein some other compounds such as formaldehyde, aldehydes, phenols, they also, which are present in the smoke, they inhibit the growth of the bacteria. So drying by smoking is also the best method of the fish preservation. Then the third one, we talked earlier about the chilling, but here in the one of the aspects is the freeze drying, that the fish are frozen, the first frozen then placed in vacuum. So they are first frozen and then they are placed in the vacuum where ice evaporates directly. It is called as a sublimation, and it preserves the color, flavor, and the nutrition, but it is expensive. So we have the three modes of the drying fish, that is, one is sun drying, other is smoke drying, and there is freeze drying. And the freeze drying is one of the best methods wherein it preserves the color, flavor, and the nutrition, but it is less expensive. So the keep in mind that the sun drying, the smoke drying, and the freeze drying, these are three best methods of the moisture removal. Then salting is one of the best preservative. Salting is best preservation methods wherein osmosis plays an important role. It draws the water out. So when we what we do have the two types of the salting, that is, one is dry salting and other is wet salting. So salt draws water out of the fish flesh. So when we sprinkle salt on the fish, so it the osmosis starts. It takes the water out of the fish flesh, and just like when you must have seen the cucumber, when you cut a cucumber and put some salt on it, the water begins to ooze out, and this creates two preservation effects simultaneously. It dehydrates the fish and creates a salty environment. Now two things, what happens: it dries fish, or we can say it dehydrates the fish, that is, water is taken out, and secondly, the salt makes the environment not good for the for the growth of the bacteria. So salt inhibits the growth of the bad bacteria, and secondly, it removes the moisture. So two main types of the salting I was talking about. One is the dry salting. The fish are layered directly with the salt crystals on the container. For example, these are the fish wherein the salt is sprinkled on the fish and the water is extracted and it forms a natural brine. And the second is wet salting. We call it as a brining as well. The fish are submerged. They are dipped in the salt solution for a set period of the time, and both play an important role in the osmosis, taking the water out and then inhibiting the growth of the bacteria. The fifth important point of the fish preservation is the smoking. I was talking about the earlier slide as well. The smoking, there are two aspects. One is the heat and other is the smoke. Two things play an important role. So smoking combines the three preservation forces in one. Three preservation forces in one. One is heat, other is drying, and the third is chemical action. So heat partially cooks and pasteurizes the flesh. So the point number one is heat, second is drying, and the third is chemical action. So firstly, it partially cooks and pasteurizes the flesh, and secondly, smoke removes the moisture from the fish surface, and thirdly, when you smoke a fish, when you use the wood or any other wood substance, what happens: the wood contains phenols, aldehydes, and organic acids, and they are the natural antimicrobials and antioxidants. Smoking is further of two types, that is the cold smoking, and we say hot smoking. So when you use the temperature 25 to 30 degrees centigrade Celsius, so fish is flavored and partially preserved. It requires refrigeration, and we can say it is partially preserved, but requires other means of the preservation or the refrigeration as well. In the hot smoking, what happens: we smoke it at 70 to 80 degrees centigrade, and the fish is fully cooked, preserved, and it can be stored at the room temperature for the shorter period of the time. So smoking at 25 to 30 25 to 30 degrees centigrade, Fish is partially preserved, but at 70 to 80 degrees Celsius. It is it is preserved fully, it is cooked fully, and it is preserved and can be stored at room temperature for the shorter period of the time. Then the sixth important method of the fish preservation is canning. So we can see that the canning industry is booming, is flourishing. It is all because what we do, that we put fish in the tight containers, in the metal containers, and we subject it to the high heat and almost up to 100 116 to 121 degrees Celsius. And we also apply pressure or retort, which kills all the bacteria in the fish in the container. And even the when we use the pressure, all the spores present in the container, they are also killed. And then the sealed environment prevents the re-entry of the microbes, that is the contamination. So here the shelf life is 2 to 5 years. It is a very good shelf life. So but at room temperature, we do have the in the markets available canned sardines, canned salmon, and which is very much in demand in the global market. And even in the India's CIFT, that is Central Institute of the Fisheries Technology, they have developed the canning protocols for our Indian species, that is the rohu, katla. So that is the what methodology we have to take care of for the canning of the katla and rohu. This method is developed in the CIFT Kochi. And the seventh method, common method, is pickling. We call it as the chemical preservation, because in the pickling what we do, we use the acetic acid, that is the vinegar. Commonly found acetic acid in our home is vinegar. It lowers the pH of the fish up to 4.5. That is wherein the most of the bacteria cannot survive. It is a acidic condition wherein the bacteria, I am talking most of the bacteria cannot survive. And the second is fermentation. It uses naturally occurring bacteria to produce lactic acid, and which use the same effect. So even in the pickling, the pH is maintained up to below 4.5, which is acidic, and in the acidic environment most of the bacteria are unable to survive. Second is the fermentation, wherein the naturally occurring bacteria produce lactic acid, which achieves the same effect, but with the complex flavor development. Here in the flavor changes. And for example, in the real world, we do have the examples as the nagari. It is a fermented fish from the Manipur. We do have the siddu nagari from the Manipur, siddu from the Himachal Pradesh, and narezushi. Narezushi, it is a fermented fish that is a sushi origin, found in Japan, Then we do have the surströmming, that is the Scandinavian method of the fermentation, surströmming. So it is a method of pickling or fermentation in the Scandinavia. Then one of the methods for the fish preservation is the chemical preservation, which is the modern method. And wherein what we do, we use the food grade chemicals and we can extend the shelf life directly by inhibiting the bacterial or the fungal growth and preventing oxidation. So the common agents which include which we use during the fermentation is sodium benzoate, or BHA and BHT, that is butylated hydroxyanisole or hydroxytoluene, butylated hydroxyanisole or butylated hydroxytoluene. These antioxidants, they prevent rancidity and in the fatty fish. And also one of the another chemicals which is used is sodium nitrate. It is used in some cured fish products. So what we find that during the chemical preservation we use the food grade chemicals which are not so injurious to the human body. We use the sodium benzoate, we use BHA and BHT, which is butylated hydroxyanisole or butylated hydroxytoluene, and then sodium nitrate is also used for the preservation. So this was all about the common methods. The first go through in the for the common methods methodology used in the fish preservation, that is the traditional as well as the we discussed traditional as well as the modern method. Next time we shall be dealing with the history of the fish preservation, and simultaneously we shall be covering on this very fish processing and preservation topic. Thank you. Thank you for watching. It is my request to you all to like and subscribe my channel Biolearnia.